Good morning, good afternoon, good evening, and good night to all my friends around the world. Just a day before Thanksgiving, we are meeting here today to listen to one of the leading experts from the pulse work in the Southeast Asia. My name is Dil Tavaraj. I am Associate Professor leading Clemson University Pulse Quality and Organic Nutritional Breeding Program. And we are here to celebrate our Going Organic seminar series. We bring experts around the world to talk to you from breeding, nutrition, and food processing. First, I'd like to thank our sponsors, the United States Department of Agriculture, USAID, Feed the Future Innovation Lab, Cornell, Clemson, K-State, and many other collaborators. South Carolina Department of Agriculture, the Good Food Institute, the Food Chart, and USDA Agricultural Research Services. Because of their generous support, we continue working on organic and other conventional pulse research. And then I like to thank Mr. Tristan Lawrence, my program manager, who makes things happen on time. Before taking much time, I wanted to introduce today's speaker. Dr. Rajeev Varshney. Most of you know by his first name as Rajeev, and he is one of the leading researcher on the genomics and the chickpea and the groundnut and many legume crops that he has been doing at the ICRISAT. Professor Rajeev K. Varshney, Research Program Director, Accelerated Crop Improvement, International Crop Research Institute for the Semi-Arid Tropics, we call it as ICRISAT, and the Adjunct Professor in Murdoch University, Australia. Professor Rajeev is an agricultural scientist specializing in genomics, crop breeding, seed system, and capacity building with 20 plus years of service in developing countries and sub-Saharan Africa, Southeast Asia. He is currently serving as the research director at the ICRISAT and the Agriculture and Food Security Chair honorary with the Murdoch University in Australia. I am sure most of you recently see the pan-genome discovery that he published in the Nature Journal, and he currently holds 19 Nature Journal articles, and he has done 12 genomes, and he's going to talk about the products that came from his program. It's about 11 products, and another Nature article is on the pipeline. And he has assembled 12 genomes and have done numerous work for the smallholder farmers around the world. It is my great honor to introduce Dr. Rajeev Varshney. Dr. Varshney, the floor is yours.
Thank you very much, Dil, for very comprehensive and elaborative introduction. Good morning, everyone. Good morning, good afternoon, good evening, wherever you are. And I'm really grateful to Dil for her invitation, and thanks to her and also Tristan for organizing the seminar. I'm very excited to be here. I just mentioned in the chat box, I'm so excited to see so many friends in this seminar. So what I will do in next 40 minutes or so, will talk some of our concept and work on fast forward breeding and rapid delivery system. So I hope all of you can see my slide. So all okay. And Dil mentioned I am coming from ICRISAT. ICRISAT is International Crop Research Institute, and we believe that all people have a right to nutritious food and a better livelihood. Our vision is a prosperous, food secure, and resilient dryland tropics. And our mission is overcoming poverty, hunger, reducing malnutrition, preventing environmental degradation. So basically to contribute towards sustainable development goals. I hope that some of you might have visited ICRISAT. If not, we would like to invite you to come to our headquarters here. In India, we have this headquarters, but at the same time, we also have our countries and research operations in 5 countries in Eastern and Southern Africa, Ethiopia, Kenya, Malawi, and Mozambique, and 3 countries in West and Central Africa, Mali, Niger, and Nigeria. At ICRISAT, we work on 3 cereal crops, sorghum, pulse millet, and finger millet, and 3 legume crops, chickpea, pigeon pea, and groundnut. Some of you in North America and the United States and Canada, you may be knowing about chickpea and also groundnut, but not pigeon pea. Groundnut is a peanut, but in India and Sub-Saharan Africa is called groundnut. So these crops, they're very important crops from the food security perspective as well as from the nutrition. But as we know that the world is facing lots of challenges, and when you talk about the international agriculture, we got lots of challenges. And institute like ICRISAT and the university system in the United States and other partners around the world with very strong support from many organizations, we all have been working to address these challenges. And I think that one of the major challenges which everyone is talking nowadays is that how to feed 9.7 billion population by 2050. And this means that if we need to do it, we need to produce 74% more food. And one of the major challenge before the scientific community or agricultural science community is that how to produce more food with less resources, less arable land, less water, less labor. And on top of that, we are talking about saving or managing this water footprint as well as carbon footprint. So I think these points, they are very, very important. And like many other colleagues, we all have been working on these aspects. And in this context, I will present some of our work. And we'll have some discussions on these 3 important legume crops, chickpea, pigeon pea, and peanut. And these crops, they are important crops for the smallholder farmers in India and Sub-Saharan Africa. And they provide that not only the food and also the nutrition, but also the income for smallholder farmers in several countries, especially in Africa, like chickpea is grown in Ethiopia, Tanzania, and Kenya. But instead of having heavy consumption in those countries, they export the chickpea to India. And you may be knowing India is an importer of chickpea from United States, from Canada, Australia, and from many other countries. So these crops are important. However, the crop productivity of these crops historically has been remained stagnant, but in recent years, things have changed. So what ICRISAT and its partners, national programs, have been working on the crop breeding and elite disciplines and targeting to enhance the crop productivity, addressing the issue of the disease resistance, pest tolerance, combating those environmental stresses, enhancing the nutrition. And while doing these things, we have the focus on the sustainability. So ICRISAT and its partners have been delivering lots of varieties in last many years. But when we talk about the modern genomics era, and we all know that there is a huge advances, tremendous progress in the sequencing technology. And if you see just last 30-40 years, it started from 80s, but in the last 10 years, this is a tremendous progress. So all people around in any field, either human genetics, plant genetics, animal genetics, they're trying to use that. And recently we talked, and we know that these technology, they are making lots of impact on plant breeding. We believe that this is the time now that both sequencing and phenotyping technologies, they are very important for understanding the genomic variation, because you can have these plant genetic resources either coming from the gene bank or breeding program, different kind of populations, and you have a possibility to undertake the sequencing. You can develop any reference genome. We believe there is a need for the democratization of the sequencing technology. We need to have the precision phenotyping technology. With the help of sequencing technology, now we can create the pangenomes. And when we have these things, data set from both sources, we can go for the trait mapping. And nowadays, not only those genes or QTLs which we have been doing last 10 years, it is possible now to undertake or to mine the haplotypes. You can identify the superior haplotypes through the pangenomics approach or GWAS approach. So I think this is one important thing. And in this direction, at ICRISAT, we have been working on different aspects. As Dil mentioned, that we have been contributing, or our genomic center has been taking the lead or contributing in assembling the genomes of several crop species, including pigeon pea, chickpea, pearl millet. And in the case of groundnut, we have the yellow tetraploid groundnut. So we initially worked together with our partners to assemble the genome for A genome and B genome. And then eventually we have also assembled the genome for the cultivated tetraploid groundnut and for both subspecies Fastigiata and subspecies Hypogea. I should also mention that initially when we were developing the genomes, at that time point, sequencing technology, they were not that advanced as we have now. So some of our genomes like pigeon pea, they were developed when the sequencing technology or Illumina sequencing was providing just 36 base pair reads. So some of those genomes at that time point, they were really very good, but now in the current scenario, they are not very high quality. So we have been also improving those genomes, and not only for ICRISAT mandate crops, we have been working with the different partners to assemble the genomes of sesame, mung bean, azuki bean, pea, longan, soybean, etc. So our center has delivered about genomes for about 12 different crop species. So as I said, so initially if you see that crops like, for instance, in the case of pigeon pea, this is our previous genome, which is not very high quality because this was based on the very small sequencing reads. But now by using the high-seas technology, we have been able to assemble the genomes in a very nice manner. And some of those misjoins and those chromosomal error or joining error, etc., we were able to fix those issues. The same thing in the case of chickpea, that we had many we had a lot of scaffolds floating. They were not anchored to the chromosome, but now we got chromosome length genome assemblies for all the different 6 species. So what I wanted to say that we keep on improving the quality of the genome so that we can have the high resolution of genetic analysis and we can use them in the plant breeding. And as we know that by using these genomes, we can have some idea about the genes, but genes do not work in silo. And if you would like to understand the spatial expression, temporal expression, then for that purpose, we have developed the gene expression atlas for chickpea, pigeon pea, and groundnut by sequencing, by doing the RNA-seq for 30 to 40 different tissues, representing different parts of plant life cycle or with the tissues which were challenged to the different stresses. And once you got those gene expression atlas, they are very helpful for your functional genomics analysis and also to understand the gene expression and mine the genes under those different QTLs. I will talk something about it later. But nowadays, when you have the reference genome, so one genome does not solve all the problems because you can have the genes only of that particular individual. So at ICRISAT, we have been trying to understand this or catalog those genetic variations for different crops, including in the case of pigeon pea. A few years back, we sequenced 300 pigeon pea lines. We tried to understand the center of origin and also that migration route, and also, by combining that phenotyping data with these sequencing data, we were able to identify that markers associated with the different traits. And then in the case of chickpea also, we have done initially 430 chickpea lines, and we have tried to identify the markers associated with the yield and drought and heat stresses, in addition to answer many biological questions. And recently, as Dil mentioned, that this paper appeared on 10th November in Nature, where we have reported the chickpea genetic variation map based on the sequencing of 3,366 genomes. So this was not planned number, but somehow this number became very attractive, 3,366. So what we have done that we have generated more than 21 terabase pair data. We have identified about 4 million SNPs. And in this data set, we had the cultivated as well as the wild accessions from the 7 different sizer species. And you can see these different statistics as we expected that in the case of wild species, we had more variation than the land races. And if you go to the breeding lines and if you go to the different chromosomes or so, you can identify that hot spot for the sequence variation, etc. And in that way, you can also identify that genomic regions which might have undergone the selection pressure. We have discussed all these things in the detail in that paper. But one thing I would like to mention that based on these data, we have developed the pangenome. And when we did the simulation study, etc., if you increase the pangenome, then you will be having the decrease in the core genome, but then there will be increase in the dispensable genome. And when you keep on moving further after this 2,500, this number, this increment is very low. So even in the case of chickpea, we got about 20,000 exosomes. We believe that based on these 3,000 exosomes, we are already reaching to a level of the plateau. But nevertheless, we and our partners, they are still interested to sequence many more genotypes and to understand that, well, if we are not missing some interesting genes. And this data set indicated that our first genome, what we had, CDC Frontier, and after that, there were several other genomes. We identified about 1,582 novel genes which were not present earlier. And we are finding these interesting genes in many important pathways which are associated with the adaptation or resistance to the different diseases or so. And not only that, we also did some interesting analysis. I don't have time to go in the detail, but these kind of things have been covered by many newspapers, media or so, and one can read about that one. But one interesting thing is that we asked this question that when on the Fertile Crescent, how chickpea was moved in the different parts, and then these colors that denote different regions. One thing our data indicates that I think that there were simultaneously two paths for the chickpea migration. One was that towards that basically at one time they were in the South Asia and North Africa or East Africa. And the second path was from the Fertile Crescent to Mediterranean, and other side is the Black Sea and Central Asia. And then through the Kabul or Afghanistan, this also came to India. And India now, this is the largest chickpea producer country. In India, we are having two types of chickpea, Desi and Kabuli. And based on that literature or so, it seemed that when the chickpea entered in India through Kabul, and which was the white colored chickpea, then they started to call it Kabuli chickpea. So a lot of interesting data from that perspective. And in terms of the size and species divergence, there were a lot of different kind of estimates, but I think this is the first time that based on the whole genome sequencing data of large number of exosomes, we were able to estimate these species divergence. So for instance, from the size of kinetum, from all these different groups, that's happened around 21 million years ago. And those two different basically the Echinosphromum was separated about 15.3 million years back. And if you see that divergence time between Sisar-Aritinum, with its closest progenitor, Reticulatum, this was about 12,600 years back. Another thing that our paper also described that many times we always talk about the good genes, which is great, but now based on this, our data set, we try to identify the genome part or genomes, that how much genome has remained under the evolutionary constraint. And then we have identified about 8% genomes, how many genes you are having. And we did a lot of analysis. And all these analysis indicated that in the case of elite varieties, so when you go from the wild to cultivated to elite, you keep on going down with these deleterious alleles. Nevertheless, even this present day cultivars also, they are still having that higher amount of those deleterious alleles. You can call them that genetic burden or genetic load. So these data indicates that we are having the target sites which can be further purged through either genomics assisted breeding or genome editing, so that you can enhance the proportion of the alleles in the genome which can have the higher crop performance and adaptation to the different environments. So you can identify those genes from that perspective. And nowadays at our lab, we are also using the approaches like systems biology. And here we try to merge or integrate these different approaches, including the transcriptome, proteome, metabolome. And nowadays we are also talking about integrating the epigenome as well. And based on these things, we would like to link with this variation and then try to identify the genes and pathways which are involved in the different metabolic pathways which can become the target for that genome editing. And some of those things we can also address through the genomics assisted breeding. And in this aspect, we are proposing the hypothesis that, well, how we should use these systems biology approaches. So we should be having these hypothesis driven. We should have the omics data integration. And after that, we need to identify or we need to link the genotype to phenotype. And we should use this thing mainly for understanding the complex trait. And we have used this kind of approach in the case of chickpea, and where in chickpea we have been targeting this drought tolerance, mainly focusing on the drought related traits in the roots. And we have done a lot of work on this aspect, a lot of work already published. I'm not talking about it here, but what we have done, that not only this genomics work recent years, we have done a lot of transcriptome, proteome, and metabolome work by using the near isogenic lines for the drought tolerance. And when we have combined all these data sets, we have identified, interestingly, the 10 proteins which are showing the correlated expression both in the transcriptome and proteome data sets. We have identified about 6 metabolites, and we are also identifying some interesting pathways. So that the thing is that not only that you should be able to address the issue through the genomics assisted breeding, but in some other crops or so, or even chickpea, if you would like to use the other approaches like genome editing, you can work in this direction. And the other trait which we targeted through the systems biology, and this was about the fertility, sterility reversion in the case of pigeon pea. In pigeon pea, we have the cytoplasmic male sterility based hybrid breeding system. We brought this thing from one another species like Cicer sericeus. But then we got some plants, and then those plants, they were very interesting plants that sometimes they were sterile and sometimes they were fertile, and this was very interesting phenomena. And then we have done a lot of work based on that plant phenotyping, physiology, and cytology. And after that, we have gone to the nth level, and we have done this transcriptome analysis and also the proteome and metabolome analysis. And by combining all these approaches, we have identified that there are some interesting things in the auxin biosynthesis pathways, and also that based on these things, when you are having that higher temperature, then those cells, they go under stress, and then you are having low level of the auxin production, and then those cells, they are having the deprivation of the sucrose. And I think that this kind of research is also very interesting from the breeding applications, because in the case of pigeon pea right now, we are having 3 lines based hybrid breeding system. And if we can identify those kind of causal factors which are making this same plant fertile and sterile, we can move towards a 2-line hybrid breeding system in the case of pigeon pea. Now let's talk about the breeding. And from the breeding applications perspective, breeders, they are always interested when we talk. And when I joined ICRISAT long back, this was always a question that breeders say that, well, your technology are very expensive and we cannot use in our breeding program. So one of the key things for us has been that how to bring the cost of the genotyping lower for both genetic analysis as well as for the breeding applications. At our center, we started with the SSR markers, and in this journey, they have developed a different kind of marker genotyping platform, DART, genotyping by sequencing, and all these things. And you can say, why so many marker platform? Because one size does not fit all. If you need to develop the genetic map, maybe the GVS approach will be good. If you need to do the GWAS analysis, maybe the 56,000 SNP array is good. If you need to understand the genome, whole genome resequencing is good. If you would like to go for the marker-assisted selection, where you need to target only few SNP, then 10 SNP panel, which we can do the genotyping in just $1.5 per sample for one line, then this screening becomes very, very cost effective. And now for the genomic prediction, we have developed the platform that for genomic prediction, you do not need more than 2,000 SNP markers in crop like chickpea. And here the cost is going about $8 to $10 per sample. So we are always interested that how we can bring these costs down. And based on these things, what we have been doing, depending on the objective, we are doing the genotyping of the biparental population, multi-parental population, germplasm collection, and we are doing a lot of different kind of phenotyping for in the field, in the greenhouse, in the lysimeter, leachy skin system, or for the different pathogens or diseases. And then we are combining all these things together, and we are moving towards the trait discovery. And in last few years, we have been successful to map 20 to 50 traits in each of these crops, chickpea, like drought tolerance, heat tolerance, salinity tolerance, ascochyta, helicoropyte. Some of these traits, they were moved further in that industrial production, like in the breeding applications. So the same thing in the case of groundnut, we got this diagnostic markers for leaf rust, late leaf spot, iron deficiency, and high oleic thing, and aflatoxin contamination. And same thing is in the case of pigeon pea, fuserum wilt, sterility, mosaic disease or so. So we got larger number of markers. And so, you know that initially I was talking about understanding the genomes and the genes. Then now I'm talking about to understand the genetics. Now the third component is, which is very important, that how we are translating these things from the genomes to the plot level. And for that one, it's really important that we are working very closely with the breeding programs and ICRISAT. We are having a really good network in different countries. Like for instance, in India, we are working with the Indian Institute of Pulses Research, Indian Agriculture Research Institute, many universities, the same thing in the case of groundnut. And not only in India, we work very closely with our national partners in Africa and many other countries in Asia. So together with them, by using the genomics assisted breeding, and then we have developed the superior lines, and our national programs, they have done the multi-location evaluation in their different target locations, or in that followed their rigorous varietal testing pipeline, and eventually they were having the successful release of several varieties. For instance, in the case of groundnut or peanut, you know that when you are talking, this is the oil seed crops. If you take seeds oil, then you are having three different type of acid composition: palmitic acid, linoleic acid, oleic acid. Linoleic acid is not good for health because this is coming from the polysaturated fatty acid. So some varieties like Sun Oleic 95R from United States is having more than 90% oleic, and this you can see that linoleic acid is minimized. So there is just a change in the pathways that if you increase that one oil composition, other will be going down. And now through the molecular breeding approaches, we have developed several lines. You can see this green color thing, more than 80% oleic acid. And in India, these varieties, two varieties through the Directorate of Groundnut Research of Indian Council of Agriculture Research were released, Girnar 4 and Girnar 5. In fact, last year, Prime Minister Modi included these Girnar 4 and Girnar 5 lines in the set of the 17 biofortified crop varieties when he dedicated to the nation on the 75th birthday of FAO. So we feel also very excited when these kind of research is recognized at that level. In the case of chickpea, with Ethiopian Institute of Agriculture Research, this variety for drought tolerance was released in the 2019. You may be knowing that in many developing countries, agriculture is done by those breeding program, public breeding sector, unlike many developed nations where you are having the industries. So in these countries, this is really important that public sector should continue to invest and to continue to work in this direction. Otherwise, you will not be having many varieties, especially in the self-pollinated crops. Similarly, other institute like IRI, we developed this variety Pusa chickpea 10216 for the drought tolerance, for fusarium wilt resistance, Pusa Manav, and University of Agriculture Sciences. Dharwad, we had that variety, and this was the Super Annigri. And similarly, we are having, in this year itself, we also had two varieties for the drought tolerance, one for the fusarium wilt. So there are lots of varieties, and this year also, when Prime Minister Modi released these, were dedicated several varieties to those nations, about 35. And in this thing also, two varieties of the chickpea were also included in this set. In the case of pigeon pea also, for the fusarium wilt or so, we have released these variety. So I think in last 3-4 years, through the genomic interventions, at least 11 varieties have been released in India, Ethiopia, and now many others. They are in the pipeline, and they are reaching to the smallholder farmers. Now, the next question is, which was the title of my talk also, that how we can make this breeding much more fast forward. And so fast forward breeding is not an approach, rather this is a framework. And we believe that now, because of those sequencing and genotyping technology, it is possible to identify large number of genes, and you can have the haplotypes. I will show in the next slide. But so based on this haplotypes, you can go for the haplotype-based breeding because you have a possibility to identify a number of haplotypes because you got the sequencing data for large number of exosomes. Now, based on this genomic data, phenotyping data, you have a possibility to undertake the genomic prediction. Recently, we have also optimized one approach, and this approach called optimal contribution selection. And in this approach, we try to identify a good set of the parents, which brings the diversity as well as which brings that good genetic gains or good traits together. So we need to make a balance. And now when you are having these systems biology approaches or different kind of thing, and if you really got the causal genes, you can go for the genome editing approach. And now for any of these approaches, and you might have heard about the speed breeding, so basically which is that meant for the advancing the generations, one needs to use the speed breeding in combination of one of those four approaches. And if you can do this thing, then you can make really fast forward genetic gains. You can enhance the breeding in much more faster manners. Now, as I said, that in terms of the haplotype-based breeding, so you have a possibility to generate the sequencing data for large number of genotypes for the different genes. You can classify those genotypes based on the haplotypes. You have a possibility to associate with the phenotype, and based on these phenotype, for instance, for this particular gene, you got 4 different haplotypes, but when you do the haplopheno analysis, you identify a superior haplotype. Like for this gene, H3 haplotype is superior. For gene 2, H1 is haplotype superior. For gene 3, H2 haplotype. So when you are having this superior haplotype, then you can ask a next question, that those elite varieties which farmers are growing, whether these varieties, they got the superior haplotype or the different kind of haplotypes. And then we have seen in the case of chickpea data set that many varieties, they are having still inferior haplotypes for several traits. So now this is the time that how we can replace those inferior haplotypes by much more better haplotypes or the superior haplotypes. And I think this is the concept of our haplotype-based breeding. And we have used this haplotype-based breeding approach and also the genomic prediction in the case of chickpeas. And as I told that we have generated the sequencing data for 3,000 chickpeas. And not only that, one of the other aspects for that paper is that we have been successful to undertake phenotyping for 3,000 chickpeas at 6 different locations for 2 different years in India. This is a huge work, and this is the coming from our breeding programs at the different locations in India. And not only that, we also have generated this phenotyping data, or we also evaluated for large number of nutrition traits. I'm sorry that I did not include that slide here, but we can talk sometimes about the nutrition trait because I know you are interested in many other colleagues. But nevertheless, we generated this phenotyping data, and many people, as we do the GWAS analysis. This is great. But not only the GWAS analysis, we went further to target the 67 genes. We identified 350 haplotypes. We asked a question that which haplotypes they remain consistent and stable across the different years. And then we say, oh, these are the superior haplotypes. But then you can ask the question whether they are really superior haplotypes or not. And we asked a question by taking about 129 released varieties in 10 different countries, and we classified those varieties in 3 different groups, like release period 1, which were the varieties which were released before Green Revolution, several varieties which were released after the Green Revolution in 90s, and the new varieties released after 90s. And now breeders, when they were developing the varieties, they were doing the visual selection. But interesting thing is, as you can see, the number of the haplotypes as they are accumulated in these varieties. So now, for instance, in that RP1 varieties, you don't have many superior haplotypes, but in the varieties of RP2, you got better number of superior haplotypes. And in these year 3 or the group 3, they got much more better haplotypes or those things. So now our approach of haplotype was validated based on those released varieties, and based on all these data sets, we identified that yes, we have some good haplotypes in these varieties, but still our germplasm got many more haplotypes which are still not integrated in these elite varieties. So our approach is, can we integrate those new haplotypes in these elite varieties, and then can make those lines further high yielding varieties. And by doing this thing, we have identified about 56 promising lines which can be used to introgress the new superior haplotypes in the breeding programs. I think this is really exciting research. The other thing is that how we can do this genomic breeding, and we have combined this optimal contribution selections. So for instance, generally the breeding program, you can keep on taking these kind of parental combinations, but now this green color line or so, and you are having this progenies, you are having the candidates, so you can really predict and you can identify that which genotype I should be taking, which can take our progeny in this direction or so. So I think that we are working on those different kind of aspects of that optimal contribution, and also that genomic prediction by using that GYE approach, and also the new approach called OGM approach, where we are also integrating the environmental coordinates. And the other approach, earlier I was talking just introgressing the haplotypes, but now here we are talking that when you have classified the genomes in the different haplotypes, and then we are estimating the breeding value based on the haplotypes. So this is the other genomic breeding approaches. And our data set indicates that if you can target even the traits like 100 seed weight, you can enhance the performance up to 23% by using these genomic breeding approaches or so. And in the case of chickpea and pigeon pea, we already are utilizing these approaches, for instance, genomic prediction in the case of chickpea, groundnut, pigeon pea, and our data set already indicate that traits like seed yield, seed weight, etc., we are having the prediction accuracy up to 0.6% or so. In the case of pigeon pea, where we are having the hybrid breeding programs, we could also predict the better parental combinations for developing the hybrids. So this is another beauty of that one. So what I wanted to say that by using these approaches, genomic approaches, breeding approaches, and by combining with the data analytics, you can make the breeding fast forward. So this is good that we need to have the fast forward breeding approaches for delivering the varieties. But we believe, and especially when we are working in the developing countries, this is not a major issue in the developed nations, but in the developing countries, especially in Africa and South Asia, farmers, they still grow the old varieties. And even if they are growing the varieties, if their produce depends on the rainfall. And if you are having the good rainfall, they will get higher produce. But even if they get higher produce, this is not translated in the revenue generation and money make in collecting more money. So we believe that when you are talking about the international agriculture, not only the fast forward breeding framework, we need to have the rapid delivery system also established. And if this is there in some countries, this needs to be strengthened. What does it mean? That when you got these elite varieties coming from any approaches, they need to reach to the farmers in the real time. And for that, we need to have the seed system really very strong. Farmers, they are still growing many old varieties. And when farmers get the varieties, this is really required that we need to have really good integration of the better agronomy. They need to be provided the access through the digital decision support tools, because nowadays everybody is having almost this mobile, because mobile technology penetration is very high. So one can provide those digital, through the digital agriculture, those decisions to the agronomy. We need to work in the direction of the reduction in the post-harvest losses. In the developing countries, you got a lot of losses in that when the crop is standing in the field. And then when you got even this produce, and then there are always the difficulties where they should sell, what should happen. So I think this is another important area that farmers need to be connected to markets in such a manner that you have minimum number of intermediary, and all of those profit generation goes to the farmers. And you can enhance their value further if you can if you can have the value addition, if you can connect them to the industries. In that way, farmers can not just generate more produce, but they can get more income as well. And for the consumers also, you will be having really good nutrition delivered to the food basket to the consumer. So I think this is another approach we need to focus more on that one. Here, I would like to mention very quickly an example that we have used some of these approaches for the tropical legumes project. This project was a big project, ran for about 12-13 years, and I had a privilege to lead this project for about 7-8 years as a principal investigator, funded from Bill and Melinda Gates Foundation, and the total investment was about $67 million. Together with the CIAT and IITA, with the 15 different national programs, 13 programs in Africa, 2 programs in Asia, 1 is India and Bangladesh. And we run this program in 3 different phases, and we had the 6 different legume crops. So as I said, so this is a huge investment and lots of partners involved in this project. But as a result of this continuous investment or so, this project delivered about 266 improved varieties, produced about 500,000 tons certified seeds, 6.1 million ton grains. And we have documented all these research and all this extension work in a special issue of the plant breeding, and also 2 books talking about that sowing legume seeds, reaping cash, or enhancing a smallholder farmers' access to the seeds. So we have documented all these things work together. And now you can see some of those impact, that how this project has made the impact. Now, if you talk about, for instance, in Ethiopia, in the case of chickpea, earlier annual average was about 1.1 ton per year, 1,118 tons per year, and this number reached to the 3,416. Same thing for the common bean. In the case of productivity, we were able to enhance from 1.3 tons to 2 tons per hectare, 1.3 ton to 1.6 tons per hectare in the case of common beans. Similar example in Nigeria, in other countries, that how we were able to impact the households. For instance, in this particular case, earlier we were just reaching 2,900, this number reached to 131,000. In the case of groundnut, 2,900 to 74,000 or so. So I think this project made really lots of impact, and we are very happy and excited to share that this kind of impact has been recognized recently, and ICRISAT for the impact of this tropical legumes project was honored with that Africa Food Prize 2021. And these kind of things again give us lots of excitement and happiness that our research is making the impact on the lives of the smallholder farmers in those developing countries in sub-Saharan Africa and Asia. So friends, in summary, I would like to say that, well, fast forward breeding expeditiously create and incorporate superior genes or haplotypes and crop improvement. A rapid delivery system needs to be established or strengthened for ensuring delivering improved varieties. We need to integrate them better with better agronomic practices. I did not talk about the mechanization, but this is also very, very much required and for harvesting the higher produce. Farmers need to be connected to the markets. We need to think about the value addition, food processing. And I think if we combine all these things, which are very complex scenario for the agriculture in developing countries, but we need to dissect them in the components and we need to address each component of that complex issue. And I think in that way, we can help farmers to generate more incomes and deliver the better products to the consumer as well. But for doing these things, when we talk about the international agriculture, this is really important that we need to have the continuous investment from the international donors, including Bill and Melinda Gates Foundation, USAID, and many other organizations in the various countries. We need to have the support from the national governments also. So I think this is very much required, and we need to continue to train this next generation of the breeders, well equipped in the using of the modern technology, plant breeding, and all these elite disciplines. So I think that's what I would like to mention. And here I would like to thank all my colleagues from our Center of Excellence in Genomics and Systems Biology, and also from ICRISAT, and all of our partners and donors and supporters with whom I had a privilege to work. And I think you can see this large number of partners, but we have been working with for the last 15 years, not with all of them on one project, but in the various time with the different projects and different activities. So thank you very much for your attention. And I would like to stop my presentation here, but will be very happy to answer any question if you have. I'm back to you, Dil. So
thank you so much for the wonderful work you're doing. It's amazing. And I'm very pleased what you're doing for the community, global food security, and it's a great impact. And audience and whoever the participants, if you have questions, please put through the question and answer box, and then I'll read the questions to Dr. Varshney. To start the discussion, Dr. Varshney, I have some, I first wanted to thank you doing this amazing work, amazing. And the question I want to start with the climate change, and you have heard about the UN Food Summit recently happened, Food System Summit, now it's called Food System Summit, in a month ago. So how do you see future with the climate change, and how we can get ready with the genome-assisted breeding and different other techniques? But nutrition also very important. How do you see connecting climate change, all the genomic resources and the nutrition, and also taking this to the smallholder farmers? This is like a big food system. How do you see this happen in India as well as to Africa?
Thank you, Dil. I think this is a very important point, very important question and a challenge. And we all know that climate change is going to make the impact. And not only that, we will be having more issues in terms of the abiotic stresses, the higher temperature drought, this is going to affect. And on that, who knows that you will be having the new types of the biotech strains and new isolates, new diseases, etc. And also all these things will impact the nutrition as well. So I think as an agriculture science community, we need to take this thing as very seriously. Now, how genomics can help? I think genomics can do not just alone, but together with the different disciplines in many ways. One of those things is that in many of these crops, either we are talking about chickpea or corn or wheat or rice, anything. Good thing is that many of these crop community, they got larger scale germplasm collection in their gene bank. Like in US, you got USDA gene bank, they got huge number of accesses, and all over the world, more than I think 7.6 million accesses they are there. Many of them, they are coming from very marginal environments. I know in the case of palm millet, when we were working, they are coming from the Sahel, where the temperature is more than 45, 42 degrees Celsius. And now through the sequencing and taking those genotypes from those extreme climates or so, and if you can combine together with the phenotyping, you may be able to identify the haplotypes for conferring the tolerance to higher heat or drought or those kind of things. So now this is the possibility to catalog those genes. Now, chickpea variety, for instance, maybe that you cannot deliver a chickpea which can survive on the 38 degrees Celsius today. But for instance, you can develop something today at 32, next after 2-3 years, you can have 33, 34. So we need to continue to make those incremental changes. But I think this is possible now.
Oh, that's wonderful. And also I have another question before I go into the audience questions. So you have released so many products, like the new improved varieties. How the seed system works? Is this accessible to the all poor farmers in India? I work with you guys a lot in India, and I know how it works. So how the system works there?
So yeah, very good point. And this is also an important point for the complex issue. So even in many countries, you already have the high yielding varieties, but they are not reaching to the farmers, especially when you are talking about self-pollinated crops, because in these countries, even you have the private sector, but they're more interested in the hybrid crops. So ideally, in these countries, you have the agriculture extension system, but sometimes this is not working. As a result, these high-yielding varieties are not reaching to farmers in the real time. But through the tropical legumes project, as I was trying to demonstrate, and now in India, we are also working with the Ministry of Agriculture on several projects, that we go to the farmers either through the formal seed system, informal seed system. We are also engaging the private sector or so. And sometimes it's also it's also very challenging to convince the farmer that if they are growing some varieties and they're always very happy with that one, they think that, well, they will continue to grow. But if you say, hey, take this new variety, they say, we don't believe you. So what you do that you need to have some farmers participatory varietal selection. We are also doing some frontline demonstration, bringing the new varieties, but their varieties, doing the farmers fairs when we are bringing the farmers from the adjoining districts. Then we demonstrate that at the time of the harvesting or flowering, which variety is good. They say, oh, this is the good one. Then we go to the State Department of Agriculture, recommend them that, well, they need to have the production of this the production of the seeds of this particular variety, and so that this can reach to the farmers. So I think this is very complex, but now things are improving. India has done a lot because they did recently very good project or initiative on the seeds. And I think if you would like to make some impact on the smallholder farmers, seed plays a very, very important role. And this is one of the important things. Yeah.
I totally agree. By working internationally, I totally agree with you. So you get some really interesting questions on the chat box. So one of the question is, in terms of haploid-based analysis, I can see this as a boon for many traits such as drought tolerance, which is true, you talked about that. But what about the disease resistance based on different plant pathogens interaction? Wouldn't haplotype-based selection of disease resistance make things easier for to overcome resistance in some cases?
Yeah, I think this is not a question, rather suggestion, because he or she is talking about these diseases. I think this is very, very good point. And I would like to say one point further, that I think for the diseases, this will be much more useful from the perspective of that evolution of those strain or so. So for instance, today you are having some, and that's the reason that when you are having even the disease-resistant varieties, sometimes you are having the breaking down of those disease resistance. So now if you can have more haplotypes conferring the resistance for particular strain or isolate for a given fungal or viral species, I think this will be good. So I'm in alignment of the points which they have suggested. Yes, this will be very, very helpful.
Yes, and now there's a question from actually one of my PhD students from India. She has worked on chickpeas, so she's very interested on improving the protein and the quality of the chickpeas. So she was asking, generally minor QTLs or alleles associated with quantitative traits are difficult to detect. Can you please provide some solution to this problem?
So yes, so in fact, way back, I think this is sometime 2009 or so, I do not remember. In the case of peanut, because in the case of chickpea, we were really very lucky. I will say that we got some major effect QTLs related to the roots, and they were providing the yield advantage when you transfer them to that elite varieties. In the case of peanut, when we did this analysis, we did not find this major QTL. We had minor QTLs or alleles. And if you would like to bring them together, it's impossible through the marker-assisted selection or those approaches. So Sonia is right. And in fact, we published that paper, and some people asked, and I wrote in that paper in the conclusion, and that this was published in TAG, I say that molecular breeding like marker-assisted selection is not going to be a useful approach for improving drought tolerance in the case of groundnut. People ask that you are a molecular person, everybody sells the approach, and you are saying that this is not going to be helpful approach here. I say, yeah, this is correct. So to address those issues, what we are doing nowadays that we are working on the genomic prediction. So what you do that basically you estimate the breeding value based on the genomic profiling across all the different parts of the genome or so. And genomic prediction, genomic selection, these approaches are going to be very, very powerful for enhancing the yield under those drought conditions. So she needs to use those approaches.
Yeah. We will approach you on some more new questions. And this is one of the questions from one of my colleague, Dr. Bandalio, is a chickpea breeder at the NDSU. Can you share a little bit about the status of implementing genomic selection into applied breeding efforts? I wonder if all plant breeders now at ICRISAT have used genomic prediction for marker selection. That's
so Dr. Bandilo asked a very good question. So what we are doing in that case of, initially we had a plan that we should be using for one breeding program or so. But eventually when we started to do this work and the prediction approaches, so initially we try to estimate or try to assess if this approach will be good. So what we are doing that instead of having just at ICRISAT, in India, those colleagues, if there are some colleagues in this, they are from India, they will understand, appreciate. So in India, for a given crop, there is a big network called All India Coordinated Research Project, AICRP, on chickpea. Under AICRP, they are having the centers at all different places in India, wherever chickpea is grown or so. So now, for instance, if you would like to talk AICRP chickpea, there are at least more than 30 breeding programs, and they are growing on multi-location testing, etc. Together with the network coordinator, Dr. G.P. Dikshit, we are deploying the genomic prediction not at one place, rather that when we are having this training population and they are doing this advancing the generation. So basically we are doing at the higher level by involving the different ACRIP location. So we already have deployed in the practical breeding program at the higher scale. But if you will ask, do you have the product right now? We don't have the product. But my colleague Manish Rurkiwal is leading these efforts together with Dr. Bhardwaj from IRI, Dr. G.P. Dikshit from IIPR. And I see lots of potential from that project. So we are doing, in summary, yes, we are deploying it, not at one program or two programs, rather at pan-India level. So that's the answer to this question.
That's very, very interesting. I have a follow-up question for Dr. Bandalio's question. So, Dr. Varshney, that now like even we developing a lot of phenotyping data for the co-collections of chickpeas. So is there like, so this GBS data, are those publicly available, or we approach via ICRISAT via you, or how is that work for the other researchers in the US and the other parts of the world?
So at ICRISAT, we are the public research institute, and we are funded from the governments of the different countries. We are international organization. All of our research is meant for freely available with public. We generate international public good. even before publication. So even this 3,000 is published now, but this is available in public domain for a long time. So in summary, yes, any data generated at ICRISAT, by ICRISAT, freely available. But if some data has been generated by some other partners, now for instance, you send a PhD student from Clemson and then you are having those lines, they generate data at ICRISAT, then we will not make it publicly available because that's your data. That's having that restriction strings from your funding agency, lots of other things. For us, any data set, anything is freely available, publicly available. Few years back, I think, so we had one PhD student from George Vandermark from the Washington State University. She visited ICRISAT, we had the data, she utilized this data, and we always do these kind of things. So we will be welcoming any student, any colleague to collaborate with us at ICRISAT, and we will be very happy to work with them.
This is one of the reasons we bring experts like you into this kind of discussion, because then these researchers in US, Canada, elsewhere can connect with you and do more, because we want impactful research. We want to have solutions for the food insecurity. So thank you, all your service. We still have more questions coming. And then again, from one of the organic expert here is asking a question about the whole genome analysis seems powerful. What environmental data are needed for the whole genome analysis? Is this as simple as using recorded temperature and rainfall? Does it include soil moisture and other data? That's from Mark Dempsey, one of my PhD students working on organic breeding.
Thank you. Thank you. Thanks, Mark. And I think my answer is, so these are the models, whole-genome model. And then if you got the data, so basically they consider GYE, genotype, environment interaction with together with markers or so. Now, if you got the data only for few environmental coordinates, this is fine, but also we would like to take as much possible. So now, for instance, if you got the rainfall and then soil moisture, this is perfectly fine. But now, depending on the traits, if your trait is heavily influenced from the soil characteristics or so, I think you definitely need to have those kind of data. If your trait is like for drought or heat and which is related to the environment or that rainfall, you need to have those kind of traits. So I think that data, so I think that depending on your trait for which you would like to do the genomic prediction, you need to have those kind of, you need to have the data sets for those traits. So those models do not distinguish or discriminate that what you are having or not, but this will be you who needs to make the decision that for your trait, which environmental data sets will be required.
That's wonderful. Dr. Varshney, this is a very interesting question. Which traditional breeding method is still competitive to the molecular method?
So Raju Nayak, I don't know that from where Dr. Raju Nayak is. So my suggestion will be, and I think this is the way that we need to move ahead now, that we should not distinguish that traditional method or molecular method. Rather, we should be talking about the integration of the different approaches. In the case, if you can measure some trait by your eyes in cost-effective manner, in faster manner, I will not push you, and I am talking the same thing to the different colleagues of mine here and other places. But if your trait, if your cost effectiveness, precision, time effectiveness can be improved by using any molecular tool, please use it. Now, so this will be my suggestion. So I would like to request you, so let's not have those discrimination or differentiation between those traditional method or molecular method. Any method which will be helpful to deliver the better varieties in faster manner, in less time, in less cost, let's use that one.
I agree 100%. 100%. That's absolutely right. And this is a question from Dr. Niraj Kumar. He's a wheat breeder. Are you working on the variety releasing steps to make it fast as you are working with ICA system? Normally that process takes a long time to release or reach to the farmers, something like similar to the question that I asked you.
Yeah. So Neeraj has been my friend. I know Neeraj well. So so what happens? There are two steps. One is developing the better lines. For that, we need to use the fast forward breeding method. So you got the line where you would say that, well, you got this line with better trait. Now the next point is to put in the varietal release pipeline. In India, we are having the system of AICRP. In Africa, you are having the different kind of systems. Some countries NPT, in some countries different thing. What Neeraj is saying that for any variety, when you are having the ICR system, so basically you need to have at least 3 years. One is this initial varietal trial, then you got advanced varietal trial 1, then you got the advanced varietal trial 2. So basically you need to have at least 3 years data set to release the variety. So what is happening that for some traits which are market preferred, industry preferred, or nowadays some traits which you already know that these varieties are good and they are not having the bad genes, in those cases, many ICRISAT programs, they already have abolished one of those required step from the IBT. So instead of having the 3 times, you can go for the 2 times. So this is happening, and many of us are working with the system that now earlier people were taking 6 to 8 years to develop the varieties, then 3 years for release, then total time was 9 years. Now for the development, you just need to maybe 2 years, 3 years, but you don't need to have another 3 years for release. But then these systems, they belong to the government. We do not have much influence on the government system, but we work with them, we work with our partners. But I can tell that all those people who are in the helm of affairs, they understand, and many times they understand, they appreciate, and as I said, in India, many of these molecular breeding varieties, they are now being released after testing just for 2 years instead of 3 years.
Okay. So that's all the time we have. I would love to continue this discussion, Dr. Rajeev. This is amazing, and it's very timely because we are going through COVID-19 pandemic. Otherwise, we will be in Clemson giving this talk. And I wanted to invite you to Clemson at some point, but going through COVID pandemic, food insecurity, all these challenges, and all the breeders around the world, and your center, we are doing a target to make better varieties, to make people's lives better. And I've seen that from your talk, how much impact it happened to the poor communities to make their lives better. And on behalf of the Clemson University and on behalf of the Going Organics seminar series, I would like to thank you. And thank you so much for participating, your valuable time. It's nighttime in India. I hope you have a great night. And tomorrow is our Thanksgiving, and I'm very thankful that you participate. Thank you so much. And thank you so much, everyone, for joining. Our next